Bismillah, Assalamualaikum warahmatullah. You're listening to Immunology lesson on innate and adaptive immunities. And this is part 1. What you've learned so far is the preparation for this topic. The learning outcome for this topic is to make you familiar with the features of innate immunity, specifically the physico-chemical barriers and physiological barriers, and also the adaptive immunity, which includes the cell-mediated and antibody-mediated responses. If you get these main features of them, you should be able to study more specialized questions of immunology by yourself. The way I conceptualize our immune system is by having 3 defensive lines. The first line of defense is the physical and chemical barriers. The second line is the physiological barriers, and these two make up our innate immune response. And the third is the adaptive immune response, which consists of 2 tactical branches, which are the cell-mediated and antibody-mediated responses. It doesn't mean all 3 cannot activate at the same time, of course, but generally, if the microorganisms can't get past the first line, usually they won't even see the third line, for example. Right? That's the basic architecture. So here are the first lines of defense. When external pathogens attempt to invade us, they have to overcome physical barriers like our skin. And if they come from food, the chemical barriers like our saliva and our stomach acid are part of this first layer as well. Remember, in one of the previous episodes, you learned that our cardiovascular system can be considered part of immunity. That might be something that young students never thought of before. So now I'm inviting you to also consider something more, something less obvious, let's say, which is that even your skin can be considered an immune organ. People who never study immunology might not realize that, but it definitely is. Your skin layer prevents probably most potential threats to your health every single day, and it has sudoriferous glands and sebaceous glands that produce substances that can kill potential pathogens, and so many other things that we take for granted. So now that you know this, you should be more grateful than before. This is an example where learning natural sciences can actually improve your spirituality as much as human sciences like theology or philosophy. When you think about the physico-chemical barriers, I want you to remember that some of them are dynamic. It means they actively move as part of the defensive process. A clear example of that is mucociliary escalators. We have this in our respiratory tract. It comprises hair-like structures, very, very tiny, called microvilli and cilia. The way they work is that when you have pathogens coming from your nasal cavity or oral cavity, the pathogens are going to stick onto these structures. Then the cilia will move in a synchronous way to escalate or push upward those pathogens. And they'll go all the way up to the nasal cavity, for example, where we can sneeze and our body can get rid of the pathogens. So that's clearly an active process for our first line of defense. If the pathogens get past that, they're going to encounter the second layer, the physiological barriers. These, generally speaking, have more moving parts than what we observed in layer 1 of the defense. The prime example of this is the good old fever. When your core temperature rises above 37.5 and stays above it, it's a good chance that your body is fighting something, that the physiological shielding is being activated. If your temperature is up until around 38, it's a low fever. When it gets to 39 degrees Celsius, you have a moderate fever. And when it goes past 40 degrees Celsius, you need to see a doctor immediately for a high fever. Your body alters your core temperature with the help of pyrogens, such as human necrosis factor, TNF, and interleukin-1. They affect the level of hormones called prostaglandin, which is not one hormone, I think, it's a group of hormones, lipid hormones, which you'll find interacting with your hypothalamus. So having a fever for too long can be quite damaging, but for the short term, having a fever is a great defensive move because number one, it initiates iron withholding, which means your body reduces the availability of iron in the environment. And that's useful because iron is a critical mineral for many pathogens. So it's sort of a logistical attack where we are attacking the enemy supply instead of the enemy themselves. The second benefit is, besides attacking the enemy supply, fever also attacks the pathogens directly by making temperature to be so hot that it's inhibitive for microbial multiplication. So they can't grow because most of them are mesophilic. They prefer normal body temperature. And thirdly, the extra heat gives your cells more kinetic energy, leading to increased metabolic rate. So they have more energy to mount further immunological responses. Part of those responses is inflammation. So you have 4 cardinal or classical signs of inflammation. Redness, because of the increased red blood circulation. Heat, because chemical reactions gives off heat in the increased blood flow. Pain comes from the pressure you have on your nerve endings. And edema or swelling because you have fluid coming out of your capillaries into the tissue area. In the literature, at times you see the 5th sign of inflammation, which is called loss of function. So you can add that there as well, especially in the more severe inflammation. So the inflammatory events go generally like this. You have a cut, like you cut your fingers accidentally while cutting your salad. What happens then is your skin epithelial layer is now breached. The pathogens get in. So they get past your first line of defense, your skin. So that event will trigger a bunch of cytokine release. Those cytokines vasodilate your blood capillaries, which means they make your blood capillaries larger. And secondly, the cytokines help make the blood capillaries more permeable, to make the wall becomes easier for your phagocytes to get through. And just like when you get a breach in a castle wall, the first thing you want to do is to get your soldiers to close that breach. In our case, the job is done by platelets. Platelets are involved in blood clotting to seal the skin wall. In the meantime, your combat units, your phagocytes, they will start marching in force towards the breach area. This involves diapedesis, which is the process of phagocytes squeezing through the wall of blood vessels. They do this to leave the blood vessels and go straight to the site of infection. And their movements are guided through chemotaxis, which simply means the phagocytes are directed by cytokines to go to the infected area. Once the phagocytes have finished the battle and the infection resolves, the phagocytes will leave the infection area. Then local cells, your epithelial cells, will work on repairs. The cells proliferate and you may develop scarring as part of this process. The cytokines in the inflammation are mainly released by your sentinel cells. Their actions can be a combination of paracrine and endocrine actions. Paracrine is when the cytokines act on nearby cells. Endocrine is when the action of the cytokines affects long-distance cells. Another minor point to remember is that these cytokines may perform overlapping functions. It means you don't necessarily have a single cytokine doing a single thing. You can have multiple types of cytokine doing that single thing. So that redundancy makes sense if you think about it, because if there's a genetic defect in one cytokine production, the whole immune response won't be compromised. Primary cytokines involved in inflammation are tumor necrosis factor, interleukin-1, and IL-6, interleukin-6. And the release of those pro-inflammatory cytokines are triggered when your cells detect PAMP, pathogen-associated molecular pattern, or when they detect DAMP, which is the damage-associated molecular pattern. So PAMPs are part of pathogens that our innate immunity can detect. DAMs, on the other hand, are part of damaged cells that our innate immunity can detect. So examples of PAMs are ssRNA, pyelin, lipopolysaccharide, Mannan, which is a polysaccharide. Examples of DAMs are HSP, monosodium urate, proteoglycan peptides, formulated peptides, histones. So PAMs are generated by the pathogen, and DAMs can be substances from our own cells, our damaged cells. Now, if you have listened to previous episodes carefully, you might think by now, hang on, I thought that was immunogen, the stuff that triggers immune response. And if you think that, that's really good. It means you are actively thinking and cross-referencing while listening to this. There are differences between PAMs and immunogens. I'll give you two of them. Firstly, PAMs are more general patterns. So you can find the same PAM in many, many types of pathogens. Immunogens are specific patterns. You can find specific antigens only in specific types of pathogens. There are around 1,000 PAMs that we have discovered, which sounds a lot, but that's nothing compared to the number of antigen, which is multiple orders of magnitude above that, over 10 million antigens. So secondly, antigens and immunogens are detected by antibodies or T-cell receptors. PAMs and DAMs are recognized by PRR or pathogen recognition receptors. Examples of pathogen recognition receptors are toll-like receptors, which we can find on the cell surface or on the endosomes. Another example of pathogen recognition receptor is retinoic acid inducible gene-like receptors. These can detect viral nucleic acid, which if you do a PhD, you have to study the specific signaling pathways that they trigger. Flashback. I don't miss that part of my experience. But it's useful because studying the pathways helps you find out whether your experimental variable disrupts that particular pathway. Anyway, CDSS, cytosolic DNA sensors, which as the name implies, they detect microbial DNA, and node-like receptors, NLRs. I can't remember what node is short for. You might come back to me later. Anyway, NLRs are pathogen recognition receptors that detect bacterial cell wall constituents. During inflammation, our body releases cytokines like histamine and bradykinin. What they do is they cause vasodilation. Vasodilation means your blood capillaries dilate. They become larger. They do that to increase the military traffic, so you get more phagocytes and nutrients, more soldiers and supplies, so we can get them quicker to the front line of infection. And symptomatically, this is going to show up as redness and localized heat in the area. The second thing that happens is the cytokines make our blood capillaries become more permeable, make it easy to get through. So that makes it easier for leukocytes to perform diapedesis, which is the act of pushing through, squeezing through the blood vessel wall, so they can get out of the blood vessels and mobilize towards the infected tissue sites. And that manifests physically as edema or swelling and pain, because that swelling presses on nerve endings that you've got in the affected area. And when the phagocytes arrive, they initiate phagocytosis. The main forces of our phagocyte army include macrophages and neutrophils, and they bring with them a chemical weapon called lysosomes. These lysosomes are their bombs. They contain hydrogen peroxide, protease, and other enzymes that are just seriously bad news for our pathogenic invaders. Now, of course, a lot of complicated stuff are happening in each millisecond of phagocytosis. And if you are interested in that, if after listening to this video, you naturally want to look up the specific processes of phagocytosis, then it's a good sign that you have the natural inclination to be a research scientist. Otherwise, if you feel nauseous when even learning the surface level of a topic, then find something else to serve the society, something else that you naturally want to look up and study deeply, even when no one asks you to. In the meantime, you can simplify phagocytosis in 4 stages. Number 1 is attachment, where they make the pathogen binds to them. Number 2 is ingestion, where the phagocytes engulf the pathogen. Number 3 is killing, where the phagocytes detonate the bomb that we talked about. And number 4 is degradation, where the phagocytes break down the pathogen into cellular pieces. So to go a bit more detail, the first step is for the phagocyte surface receptors to bind to the pathogen's surface receptors. That binding attaches the pathogen to the phagocyte's plasma membrane. Then the phagocyte engulfs or takes in the pathogen. So it does it by extending its pseudopods, like extending its arm out to hug the pathogen. So that engulfment produces a phagosome. A phagosome contains the pathogen. Now, the phagosome will merge with lysosome. The lysosome contains all those chemical weapons that I mentioned before. That merging forms phagolysosome. Here, the hydrogen peroxide and the enzymes that were in the lysosome, they will kill the pathogen and break them apart into pieces. And some of those pieces are released and some are kept by the phagocytes to display on the MHC, as I mentioned in previous episodes. Now, microorganisms can counter the phagocytes in a number of ways. For instance, Staph aureus possesses carotenoid pigments to shield them against hydrogen peroxide. Mycobacterium tuberculosis does the same thing, but with its glycolipid cell wall. Other pathogens, instead of just shielding themselves, they actually fire back. Streptococcus pyogenes, for example, it's got leucocidin, which is a cytotoxin, and it shoots that cytotoxin to kill the phagocytes. And other bacteria are not shielding themselves and they don't fire back. Instead, they dodge the attack by having capsular materials around them. So phagocytes can't bind their receptors to the bacteria. An example of this would be Streptococcus pneumoniae. Another defensive method that our innate immunity has is by using antimicrobial proteins. So the first example is the complement system. What you have here is a set of dozens of proteins. It can be up to 30 proteins. These proteins are coordinated and cause a cascade reaction. A cascade reaction means you've got the first protein activated, then that activation leads to the activation of the second protein, and then the third protein, and then the fourth protein, and so on. If you want to look into more details, you can compare the 3 complement pathways, which are the classical, alternative, and lectin pathways. But for this course, for illustration, I'll just talk about the classical pathway. So this one starts with the protein called C1, attaching itself to the pathogen. It does this with the help of antibodies. Remember I told you the line between innate and adaptive immunities is, what would you call it, fluid. They're not mutually exclusive. So this is a good example. Here we have antibodies who are regular soldiers for our adaptive immunity, assisting the innate immunity through the complement system. So once the C1 protein is activated, it consequently activates a series of proteins after it. C4b, C2a, all the way to C5b67. So that's a cascade reaction. This protein complement response helps with the inflammatory process. It also helps the phagocytes to move towards the sites of infection through chemotaxis. It triggers histamine production by mast cells, which leads to increased permeability for diabetes. It also aids phagocytosis. The complement proteins bind to the pathogen, which help the phagocytes to stick to the pathogen much better. And typically, at the end of it, you have a MAC attack, which is the membrane attack complex formation. So this complex is made up of an assembly of complement proteins, which will punch a hole through the pathogen's membrane, which causes the cytoplasmic content to leak out. The pathogen essentially bleeds out, and that eventually kills the pathogen. When it comes to viral infection, you're going to come across interferons a lot. The most common of them are interferon alpha, beta, and gamma. Interferons alpha and beta are great against viruses, and interferon gamma is also important in fighting cancer cells. There are other interferons, omega interferon, epsilon interferons, which you can look up later. So when you have a viral infection, typically the virus will invade our cells and release nucleic acids, either RNA or DNA. The interferon genes will then sense those foreign nucleic acids. So those genes got activated, which leads to the synthesis of interferons. And those interferons will be released to the neighboring cells and sort of as tactical warning signal, telling others that the enemy is here, the enemy is here. And then when the adjacent cell receives the interferons, because of this warning signal, it will have enough time to produce antiviral proteins. So by the time the virus finished attacking the first cell, by the time they get to the second cell, the antiviral proteins are already there. And those antiviral proteins will disable or even destroy the viral nucleic acids. So that's generally how it works. So we can't save the first cell though. They sacrifice themselves in the battle, which is not an issue if the dose of infection is small. But if the amount of virus is a lot during the initial infection, then the virus can do so much damage before our body can synthesize the antiviral proteins. But most of the time we win. Many pathogenic microorganisms need iron for their enzymes activities. So one strategic way to fight them is to attack that iron supply. We do this through iron-binding proteins such as hemoglobin, lactoferrin, transferrin, and of course, it doesn't always work all the time because some pathogens have countermeasures called siderophores. So they chelate the iron. They bind the iron to keep the iron away from our iron-binding proteins. And finally, our immunity can also deploy antimicrobial peptides. So these are short strands of peptides, rarely go beyond 50. They will insert themselves into the plasma membrane of the pathogens, punch a hole in the membranes. And that causes cytoplasmic leakage. So the contents of the cytoplasm leave the pathogen and the pathogen shrivels and dies. All right, that's it. Next episode, we'll look at what happens if the pathogens manage to bypass these two lines of defense. Until then, talk to you again. Barakallahu li wa lakum. Wassalamu alaikum wa rahmatullah.